speaker this evening is going to be Mike Farley and Kathy McCormack, and those two are teaming up to present the tracker project that we have going on. This will be a kind of a preview for them because they're going to be also presenting this at the annual conference. So with no further ado, let's see if Mike Farley and Kathy McCormack can take over the screen.
Hello, everyone. I would like to welcome you to tonight's presentation on our Goodwater Nature Tracking Project. We began as guinea pigs for the camera loan program of the Texas Parks and Wildlife Department's Texas Nature Trackers. And along the way, we have become contributors of citizen science. Now, if by chance you saw our Texas Conservation Symposium presentation earlier this year, tonight's first 10 minutes will be very similar, but please bear with us while we bring everyone else up to speed and then provide the current status of our project. We will give you some background information first, and then summarize our phase 1 and phase 2 results, share some of our lessons learned and suggestions for how a chapter can start their own game camera project, list our major contributions, and wrap up with our future plans. The Texas Parks and Wildlife Department hosts the Texas Natural Diversity Database, a statewide database of natural resources that is used for conservation planning and research. They also run the Texas Nature Trackers program, which allows citizen scientists to contribute data on plants and animals through the online iNaturalist platform. The 1,300 species in Texas that are declining or rare are considered the species of greatest conservation need, and iNaturalist data for these species that meet certain criteria can be moved into the Texas Natural Diversity Database. To support iNaturalist data collected by Texas Master Naturalists, the Texas Parks and Wildlife Department started a camera loan program. Applications targeting SGCN mammals are given priority, and all data, including observations for birds and herps, are to be shared with the relevant Texas Nature Trackers iNaturalist projects, that is, the Mammals of Texas, Birds of Texas, and Herps of Texas. As most of you know, Texas Master Naturalists are trained volunteers that provide education, outreach, and service dedicated to the beneficial management of natural resources and natural areas within their communities. There are 48 Texas Master Naturalist chapters across the state. I am in the Goodwater chapter in Georgetown, and Kathy is in the Capital Area chapter based in Austin. In 2019, the Goodwater chapter Master Naturalist was the first to submit an application to the camera program with swamp rabbit, Civilagus aquaticus, as the SGCN mammal target. In phase 1, 10 Reconyx Hyperfire HC600 cameras were set up at over 50 different sites in 3 locations: the Galt site, Berry Springs Park and Preserve, and Rivery Park. Note that the HC600 model takes only day and night still camera observations, that is, no video camera observations.
The Galt site is located on the Bell and Williamson County border, and Berry Springs Park and Preserve and Rivery Park are located in central Williamson County near IH-35. Note that project cameras were moved around at all 3 locations as conditions warranted, and the following 3 maps show the camera sites through December 7th of last year. Mike, could you put it back in slideshow mode?
Yep.
Well, I cannot keep it in slideshow mode. I can't see the, let's see, slideshow. From current slide, we'll try it that way. Resume slideshow. All right, well, let's see if I can advance. No, Mike, you're going to have to advance it for me. I can't move it.
All right, no problem.
Okay, so here's a map showing where 3 project cameras had been deployed at the Galt site. The Galt site is private property managed by the Galt School of Archaeological Research. Buttermilk Creek runs across the northeastern portion of the property. Next. Here's the map showing where up to 10 project cameras and Mike's personal camera had been deployed at Berry Springs Park and Preserve. Berry Creek runs along the southern border of the park. Dry Berry Creek runs along the northeastern border, and they converge at the southeastern tip of the park. IH-35 cuts through the western portion of the park just north of its intersection with Toll 130. Note that the northwestern and southeastern sections of the park are not normally open to the public, but the Williamson County Park Manager gave us permission to collect data there for this project. Next. And here's a map showing where 1 project camera and Mike's personal camera had been deployed near Rivery Park. The North Fork of the San Gabriel River runs along the eastern border of the City of Georgetown Park and then flows under IH-35. These pictures show a Phase 1 game camera, the custom stand that Mike made for each of them, and how the cable lock was used to secure the camera. Mike created a specific project in iNaturalist for Phase 1, which originally collected just the mammals observed with the game cameras or while we were out in the field. But later, other wildlife was also included at the suggestion of our Texas Parks and Wildlife Department contact. The iNat project made it easy to keep track of the observations, species, and observers, and Mike even experimented with making a project journal post.
This slide summarizes the Phase 1 mammal observations. We had 27 species and over 2,200 observations, with raccoon, fox squirrel, possum, armadillo, and white-tailed deer as the top 5 species. Note that most of these observations were made with the game cameras, but not all. Since swamp rabbit was the project's Phase 1 SGCN mammal target, we will now provide some background information about that species and have some details about those observations. Swamp rabbit is an endemic species in the U.S. and ranges across the southeast. It is secure in the southern part of its range and declining in the northern part. The overall current population trend is decreasing, and we are on the western edge of its current range here in Williamson County. Our 2 locations with confirmed observations can be seen just outside of this IUCN Red List range map. Silvolagus aquaticus is the largest North American cottontail at 3 to 6 pounds and 18 to 22 inches long. Field marks include a cinnamon-colored eye ring, brownish-gray fur with black streaking, and it has a hind foot length greater than 100 millimeter. Swamp rabbits practice coprophagy, which is a re-ingestion of fecal droppings, just as other cottontails commonly do. The photo shows the difference between the first digestion pellets on the left and the second digestion pellet on the right. Swamp rabbits are associated with special habitat factors such as a dense, brushy thickets and vines and wooded floodplains along lakes, rivers, and swamps. It commonly seeks water to escape danger. Fallen logs and other flood debris are used as latrines and are characteristic for this species. The latrine logs communicate information about territories, resource availability, and breeding status. Swamp rabbit had previously been documented at Berry Springs Park and Preserve, but only trace evidence, that is, a latrine log, was observed there during Phase 1 of this project. However, it has been well documented near Rivery Park, where we found multiple latrine logs with extensive and ongoing use and captured the swamp rabbit with both still and video camera observations. This map shows where swamp rabbit latrine logs were observed near Rivery Park during phase 1 of the project. Here is one of the latrine logs near Rivery Park. There's a lot of research in what constitutes a preferred type of latrine. Common descriptions typically include rotten logs or stumps that are somewhat flattened or sometimes moss covered. This is a general list of plants observed in the swamp rabbit habitat at Rivery Park in a subjective order of abundance. Here's a nice image of swamp rabbit, which inspired me to build some 100mm scale bars to attach to the sides of the logs to use in comparison to the hind foot length. The scale bars later confirmed the hind foot length as being greater than 100mm. Here we have a series of 3 images of a swamp rabbit jumping onto a latrine log taken with one of the project cameras. The following videos were taken with my personal game camera. The first video is of the same previous log, but from a different angle. Watch closely as the pellets drop and a characteristic shake occurs just before the rabbit exits the log. Those flashes that we see are from the project camera on the right-hand side. These are infrared light and they're not seen by the animal. The video is recording in infrared, so it captures that light. This video is of a different latrine log. It shows pellets dropping into small crevices in the log at the beginning and at the end. In this last video of a swamp rabbit on yet another log, there's a quick pellet drop and a shake, followed by a light snack of Japanese honeysuckle. Whenever we see pellets dropping in videos, we have made an effort to collect them as quickly as possible for DNA testing. I want to say that I'm a huge fan of iNaturalist, and after several years, you begin to make acquaintances online. Eventually, you meet some of these people in the real world. I'm very grateful that my iNaturalist friend, Dr. Russell Fowl at Tarleton State University, agreed to test for swamp rabbit DNA on the outside of these pellet samples. This phylogenetic tree is derived from the alignment of 4 pellet DNA sequences noted by their iNaturalist observation IDs, plus 4 reference sequences from GenBank, an online DNA database. The DNA sequences from the pellet samples grouped with those of swamp rabbit, not eastern cottontail, or Silvolagus floridanus. Below the tree is a DNA sequence alignment from which the tree was derived. The 4 pellet sequences and the 2 swamp rabbit reference sequences are identical to one another, but very different from the 2 eastern cottontail reference sequences. This highlighted iNaturalist observation is from Berry Springs Park and Preserve, and it was the first trace evidence of swamp rabbit found during the project. Dr. Fowl provided instructions for adding the DNA sequences that he generated to the iNaturalist observations. This is that same observation from Berry Springs with the sequence added in the DNA observation field. Late last year, we submitted an abstract about a project to the Annual Texas Conservation Symposium hosted by the Williamson County Conservation Foundation. On January 10th, 2020, we presented these phase 1 slides, and we hope to give an update on the project at next year's symposium.
We'd like to show you some additional critter photos from phase 1. And Mike is going to have to page through these for you.
Ready?
Yep. So here's a wild boar, also known as feral hog, at the Galt site. And another one a month later. And another one a month after that. Here's a gray fox at the Galt site. And another one 5 weeks later. Here are 2 river otters at Berry Springs Park and Preserve. Here is one 2 weeks later. Here's another one the next day. And 4 months later, there were 3 river otters in front of the camera. Here's a coyote at the Galt site. And another one at Berry Springs Park and Preserve. Here's an American beaver at Berry Springs Park and Preserve. And 4 months later, we have a cool sequence of beaver photos. You can tell that the beaver is somewhat aware of the camera. And the next day, here he comes. How often do you get to see a beaver gnawing on a tree? So very near to where that camera was 1 week later. And here he goes again, back to work. That's a very busy beaver. So here's a gray fox at Rivery Park. With prey in its mouth, with prey in its mouth. And here's a ringtail at Rivery Park. So now we have a series of very cool bobcat kitten images from Berry Springs Park and Preserve. Here's one bobcat kitten. And there's 2 bobcat kittens. An hour later, there they are. And here's one rubbing past a tree, just like a domestic cat. And now we have 3 bobcat kittens. You can see the eye shine in the center back. There's 2 kittens wrestling in the back right. Good luck, little guys. We weren't expecting to find this, but Mark Nuccini identified this as southern flying squirrel when he added the observation to iNaturalist. Williamson County is at the far western edge of southern flying squirrel's range. 2 weeks later, there were 2 squirrels. A month later, there were 3 squirrels. And it looks like we're going to have more squirrels in 40 days. This map shows where we documented southern flying squirrels in a 7-acre area over just 2 months of observations at Berry Springs Park and Preserve.
We're now going to tell you a little bit about the project's Phase 1 effect on the chapter and its members. During Phase 1, there were 167 members in the chapter, and 20 of them participated in the project. There were over 770 total project field research hours from July 2019 through January 2020. At a rate of $23 per hour, this volunteer service equates to more than $17,000 for our community and the state. In addition to several members purchasing their own cameras, the chapter ordered 7 more cameras for the purpose of continuing the search for species of greatest conservation need, and the 10 Phase 1 project cameras were returned to Texas Parks and Wildlife Department. 3 of the 7 cameras were funded by 2 project member donations, along with 1 corporate match. Special thanks to Randy Spurlock and Dale McLean. After some discussion, the target SGCN mammal for Phase 2 was set at eastern spotted skunk, Spiligali putorius, and its plain subspecies. The western spotted skunk, Spiligali gracilis, is not an SGCN mammal. On the left is a wanted poster for all observations of spotted skunks statewide from Robert Dowler of Angelo State University. On the right is a photo from an iNaturalist observation of an eastern spotted skunk captured on a game camera near Killeen, displaying its unique warning posture. And I would add, this was taken January of this year. Due to the COVID-19 pandemic, however, Phase 2 of the project quickly shifted to focusing on more of a basic inventory of Southwest Williamson County Regional Park with spot checks in a few additional locations in order to continue collecting observations of any kind during these trying times. Williamson County Park staff also requested that we limit the number of people during each outing to the park. In Phase 2 so far, 6 Reconyx Hyperfire 2 cameras and 1 Reconyx Hyperfire 2 Pro camera have been set up in almost 90 different sites in 6 locations: Berry Springs Park and Preserve, Lake Georgetown Stilling Basin, Rivery Park, River Oaks Preserve, San Gabriel River Island, and Southwest Williamson County Regional Park. Both Reconyx Hyperfire 2 models take day and night still and video camera observations. The standard model has a minimum focal distance of 10 feet, while our professional model is custom focused to 15 3/4 inches. Why was the professional model ordered? Many of the small mammals in our Phase 1 were not being identified in iNat, and we wanted to try to document them better. In addition, the custom focus camera can be used in other situations. For example, microhabitats, reptiles and amphibians, as well as insects.
Due to the COVID-19 pandemic, the ArcMap software used to create the Phase 1 maps could not be accessed. So the Phase 2 maps were created using Google My Maps. They showed the camera sites through September 7th, earlier this month. You are already familiar with the description of Berry Springs Park and Preserve from Phase 1. Similarly, Rivery Park was also described in Phase 1. The Lake Georgetown Stilling Basin is the outflow area for the 1,300-acre lake with a 50-foot cliff face on the south side. River Oaks Preserve is a private 6-acre developer cast-off property owned by the Texas Land Conservancy, next to a business park on the South Fork of the San Gabriel River. A camera was also placed on a small vegetated island in the San Gabriel River, about 2 miles downstream of Rivery Park. The Southwest Williamson County Regional Park encompasses 800 acres in a rapidly developing part of the county. There are 2 karst preserve areas located within the park, both of which contain gated and non-gated caves, and we received permission from Williamson County staff to set up cameras at some of these cave entrances. From February through Labor Day of this year, cameras were placed at 51 sites within this park. Mike created another specific project in iNaturalist for Phase 2.
This slide summarizes the project's Phase 2 mammal observations so far. We've had 25 species and over 1,300 observations, with raccoon again being the clear leader, followed by possum, armadillo, cottontail, and southern flying squirrel from early in Phase 2 as the top 5 species. Again, note that most of these observations were made with the game cameras, but not all. We're going to show you some images from the mammals observed at Southwest Williamson County Regional Park. Here's a bobcat, and 4 months later, another bobcat. Here's a group of wild boar and a gray fox. Here's a ringtail at a non-gated cave entrance. And 7 minutes later, here are finally 2 coyote pups at a gated cave entrance. This is the bucket cam that I built that uses the close focus camera in order to help with the identification of rodents. There are scale bars and seed containers inside. It has small entrance holes on the side, and the bucket cam was secured to prevent raccoons from turning it over to get the food. Bad things happen when you feed the wildlife. So now we generally only use scent baits, mostly cooking extracts, to attract critters. The track enclosure also uses the close focus camera. A critter walks through the carpenter's chalk and onto the contact paper on its way to and from the scent container in the back of the enclosure. The close focus camera is mounted in a modified stand and aimed straight down. An inaccessible food bait box can be secured at the back of the track enclosure, and fur snags on the walls can collect samples for DNA analysis when the target species is confirmed to be in the area. On the left is how that looks to a critter inside the enclosure, and on the right is opened up for processing. This ringtail photo on the left is from the close focus camera, and his tracks are on the right. And here we have a possum photo on the left and his tracks. And here's a photo of probably a hispid cotton rat on the left and his tracks. The track plate is on the left. It is easier and cheaper to produce and transport than the enclosure. On the right is a photo showing the 3 cameras in action. The track enclosure with the close focus camera is in the back. My personal camera and the standard game camera are watching the track plates on the right. The raccoon photo on the left is from a standard game camera. The raccoon is smelling the scent canister and leaving his tracks on the contact paper. The photo on the left shows some initial tracks on the plate. When the plate is ready to be processed after several days of activity, you take photos of the tracks with a scale marker for the iNaturalist observations and then remove the plate. The photo on the right is a final documentation of the track plate just before the contact paper is replaced.
Now we have a few more photos of critters seen during phase 1 and phase 2. We'll start with some snakes seen at Berry Springs Park and Preserve and Southwest Williamson County Regional Park while we did the camera's SD card swap every week. Here are 2 different western diamondback rattlesnakes. The 2 photos on the right actually show the snake drinking water. Here's a Texas coral snake with the red and yellow bands next to each other. Remember the rhyme, red and yellow kills a fellow, red and black, friend of Jack. So when the red and yellow are next to each other, that's the coral snake. And when the red and black are next to each other, that's probably a milk snake. 2 weekends in a row at the same site, a plain-bellied water snake was found in the camera's security enclosure. An eastern patch-nosed snake is in the top left, and a rough green snake is in the bottom right. Here are 2 photos of a black-necked garter snake. And here are some birds seen at the Galt site. Wild turkey, red-shouldered hawk, a barred owl, and again the next day. We had 1 bird at the Lake Georgetown Stilling Basin. This photo of a hermit thrush is from the close focus camera during spring migration. Here are some birds seen at Berry Springs Park and Preserve. An eastern screech owl, a greater roadrunner, an American woodcock, and this photo of a mourning warbler is also from the close focus camera during spring migration. And finally, here are some birds from Southwest Williamson County Regional Park. Black vulture, wild turkey, greater roadrunner, and these 3 roadrunners were at an artificial water drip that had been set up a month later in August.
Videos can provide more information than still images, including interesting behaviors. Links to the following 10-second videos were included in the appropriate iNaturalist observations. Here we have a ringtail with prey. And here we have mating southern flying squirrels. This is one of my favorites. This is a nest building 9-banded armadillo. He is taking leaves down into a small cave entrance. Here we have a mama possum with 6 joeys in tow. She has 4 on her back and 2 following. Here we have a striped skunk rubbing its head on the scent bait, a lot like catnip. And we also have a Virginia possum also rubbing his head on the scent bait. You see the ground moving, it's because it's crawling with granddaddy long-legged spiders. And here we have a common raccoon eating those spiders. There's also a spider on the camera.
Here are the Phase 2 numbers for the chapter and its members. During Phase 2, there were 176 members in the chapter, and 16 of them participated in the project. However, only 4 members have been consistently active during the pandemic. There were over 480 project field research hours from February through August of this year. At a rate of $23 per hour, this volunteer service equates to more than $11,000 for our community and the state. Now we'll change gears a little bit and review some logistics for those that might want to start their own game camera project. These are the items in our field kit that we carry with us each week. It includes SD cards, keys, scent baits, replacement batteries, and various gloves, bags, and other hardware. Before COVID-19, a large number of project members usually participated in the weekly walkabout. This meant that each of them could be assigned a specific role in the field, and those that would be submitting INET observations took an SD card home with them and brought it back the next week. During the pandemic, one person was assigned to each camera, and they were the only ones that touched it. The few participants in the field performed several of the other roles, and participants at home received each week's folder of images and videos from a dedicated Google Drive account that had been created. So focusing on the last item in the list, files uploader to Google Drive, during Phase 2, 2 free Google accounts were set up that come with 15 gigabyte of cloud storage, and the Drive portion of those accounts is used to share game camera files. Team members get an email with a link to the files that they can download and process into iNaturalist. This process was developed due to COVID-19, but it has turned into an opportunity that will continue even after the pandemic is over, because it facilitates accessibility for others in the chapter.
Here are some of our project's overall contributions so far. 3,551 mammal observations in Mammals of Texas project in iNaturalist for 30 species. 41 swamp rabbit observations in iNaturalist, including DNA. 15 North American river otter observations in iNaturalist. A number of bird and snake observations have also been added. 2 locations with confirmed swamp rabbit observations, Berry Springs Park and Preserve and Rivery Park, are just outside of the IUCN Red List range map. The range of Atwater's pocket gopher and southern flying squirrel in iNaturalist were expanded into Williamson County. Thorough surveys of Berry Springs Park and Preserve and Southwest Williamson County Regional Park habitats were conducted throughout the seasons. And finally, eastern spotted skunk and its plain subspecies have not yet been observed at 138 sites in Williamson County where game cameras have been set up. However, negative findings are as valuable data to researchers as positive findings. A summary of phase 1 was also presented at a local conservation conference. Our future plans include continuing periodic monitoring for the phase 1 SGCN mammal swamp rabbit, attempt to partner with River Park property owner in order to protect this habitat, support Dr. Fow in preparing a scientific article about the swamp rabbit, monitor other locations within the county, parks and preserves, chapter member properties, attempt to partner with additional conservation organizations. We plan to continue searching for spotted skunks, and we hope to figure out the right settings for our close focus camera in order to use it in aquatic environments. We'd also like to incorporate a donated drone into the project. Special thanks to Mike Finn and Amy Flynn. Contact me if you're interested in helping in the field. And we hope to, went too fast. We hope to figure out the right, contact me if you're interested in helping in the field. I will help provide driving directions for you and work you into rotation schedule as conditions allow. Contact Kathy if you're interested in helping at home. She will provide special requirements for uploading project observations into iNaturalist and work you into the rotation schedule as files become available. Hopefully you will be attending the iNaturalist 101 session at the upcoming Texas Master Naturalist Virtual Annual Conference if you are new to Citizen Science Online Network. We look forward to the day we can get together like we did so long ago in the beginning. We really miss those times. We would like to acknowledge our supporters, Williamson County Parks and Recreation staff, Susan Blackledge, Mark Pettigrew, Gary Boyd, Goodwater Master Naturalist board members, Charles Grimes, Wayne Roden, Nancy Phillips, Randy Spurlock, Bob Waring, and Jim Haley. Lots of Goodwater Master Naturalists help. Thank you. Our subject matter experts, Tanya Hamayun, Russell Fow, and Zachary Adcock. We've provided contact information here. We have a number of references. Thank you. Are there any questions?
We have a couple of questions, Mike, and would you mind backing it up to your contact information? Leave that up there while we go into the questions.
No problem.
All right, so Kate Knight asked, you guys already have some projects planned for spring 2021?
We'd like to get into other parks. We're interested in looking at Gary Park, and as River Ranch becomes available, we're interested in there. I would like to do more than we have with the Eastern Spotted Skunk. I was kind of late in getting that selection made, and then something pandemic showed up and messed things up. So we need to do more with the Eastern Spotted Skunk. We don't want to ignore swamp rabbit. We want to keep looking for that and collecting scat. I was down there today looking. The southern flying squirrel, while he's not a species of concern, we want to learn as much as we can about that. And they were there heavy in November and December and January and February. They're not there right now. And so I want to understand where they're going. and are they coming back? Let's see. I don't have any other things I can think
Sounds
of.
like you got lots of plans, hopefully.
There's plenty of work left on the table.
Great. Linda Kahn asked, well, how do you guys determine the worthwhile sites to place the cameras?
Well, that's just sometimes hit or miss. A lot of reconnaissance work. We walk a lot of sites. You can see critter trails. They're not hard to see, but we're generally not looking at the larger critter trails, but we're looking for smaller thickets and bushes and brush like that. It was just a fluke that we found the southern flying squirrel. We were in that for a thicket, and we happened to be aimed at the trunk of the tree, and that happened to be their playground. And so that was a whole new thing for us. We like riparian zone. We did that a lot in phase 1. We'll continue to dabble into that occasionally. How do I choose a site? Thank you for coming tonight, Linda Jo. She's from the El Camino Real chapter. She's also very popular on iNaturalist.
Sounds more like it's a kind of a black art, not necessarily a science, picking the spots. One of the anonymous attendees asked, who would I contact if I wanted to get camera set up on my land? And I noticed that you guys mentioned private land. So would one of you guys be the contact?
Yep, that would be me. And me and somebody else would come out and look around. We might bring a camera. We'll see what it looks like.
Okay.
The things we look for are safety of the camera not being stolen. So hopefully you got some acreage. We're looking at different habitats though.
Great. Amy Flynn asked, do you feel like you guys have been especially challenged by COVID? It seems that you're getting amazing results, and what will assist to continue the project?
Well, it is a challenge, and it was kind of a very dampening start of our second phase of our camera project. And so it slowed us down in the beginning. We're doing better now. We've adapted. It's different, smaller groups. I really miss, we would have a dozen people show up and we go out and we have a lot of fun, and we just don't do that now. The animals are still doing the same thing they did before the pandemic, so that hasn't changed. iNaturalist, I believe, is as popular as ever because of the pandemic. Let's see. Kathy has come up this wonderful thing with the sending the SD cards to people with the Google Drive. So that was something we didn't have before. Not that I want to thank the pandemic, but that's how it got here. Thank you, Amy.
All right. Sherry Wiseman asked, have you guys considered Divine Lake Park in Leander?
Well, there's a lot of herp observes amphibian watch out at Divine Lake, and there's a small wooded section in there, but there's also a lot of people that go out there. And so to put a camera inside of that wooded area in Divine Lake, it's risky. Whenever I do attempt a risky camera behavior, it's very short-lived. I'm not there more than a night or two in that sort of public environment. There's also an issue of homeless people that put tents out in those places, and there are homeless tents in those woods. I was going to say something else about this. Recently,
so public places sound like they can be difficult.
They can be. Now, I've been fortunate. I've never lost a camera, but I've had a few occasions where people have seen my camera, and I've been lucky. Young men paddling their kayaks past me and give me a nice salute, thank you for leaving my camera alone. I had Boy Scouts find a camera once, and they were just curious, and they left it alone. I've had some other encounters that I won't talk about, but people find the cameras, they do strange things with the cameras. So we hide them as well as we can. We camouflage them. And that's all we can do. Thank you.
So, yeah, Carol Travers asked, are the swamp rabbits hunted?
They're very
they're
hunted.
the large rabbits.
They're very hunted elsewhere in their range. Or at least they were historically. Whether they are now, I'm not sure. But certainly they were overhunted, or I'm gonna say they're not, they're secure in the southern range, and it's very popular to hunt swamp rabbits because they're such a tremendous foe for the hunters. They zigzag, their dogs are clueless chasing these zigzag rabbits that wind up in the water and swim away and hide. So yes, overhunting can be a problem, but the greater problem for swamp rabbit is they are habitat specialists. They require a unique landscape in order to survive. And so when populations increase and that habitat becomes fragmented, then their gene pool kind of gets a little small, and then you might have inbreeding. And so that's not good for a species. So that's the greater problem swamp rabbit has that eastern cottontail does not have, since they are a generalist and they don't need any specific habitat to survive. So hunting is just one of the problems.
Okay. And April, I believe, mentioned, since she's one of the members from the group there, that all the observations or locations are obscured upon iNaturalist to try to kind of keep the cameras and mammals safe, right?
Yes. Anywhere we have a camera, we have obscured that location. If we photograph other wildlife while we're out there, we obscure those if we're off the trail. So that's the reason a lot of my normal basic iNaturalist observations are obscured, because I have cameras in the area where I plan on going back with those cameras.
And for those that aren't aware of what exactly obscuring means, the default for an iNaturalist observation is that it's open, which means you and everyone else in the public that goes to iNaturalist can see exactly where that point is on the map. But if you obscure the observation, you and only the curators, which tend to be professionals, people that are helping out with the identifications, only the curators of that project, in this case, Mammals of Texas, can see the exact point on the map. Everyone else sees a random dot in a random box on the map. So that's what obscuring means.
We have a comment in the chat there from Rachel Hagen. She says she manages the parks, trails, and endangered species preserves out at Brushy Creek Mud, and would really love you guys to come do some sampling out there. She left her email, so I will pass on that to you later, Mike.
Cool. I was hoping that would happen.
And then we had another question come in from Jack Cochran asking, do these swamp rabbits hybridize with cottontails?
No, they do not, according to an article that Dr. Fowl told me about, that people have done transects that go through both areas where the species are known to live, and collected either, I don't know if there was live samples of mammals or the scat or what, but none of the collected samples showed hybridization.
All right. I don't see any current questions anymore right now, so some thank yous from everybody. And that was actually from Tanya.
Thank you, Dr. Tanya.
All righty, I think we're going to call this to a close. Mike, Kathy, awesome job. Very, very impressive. Very pleased with your presentation. I hope we can have you back when you have more to present, because it was incredibly awesome. For everybody else, we've got Chris Maxwell Gaines coming up in October. He's going to finish his water presentation. This is going to be more on developing your own in your home water recycling program. Other than that, I think everybody have a great evening. And again, Mike, Kathy, kudos to you all. Great job. Thank you so much.
Thank you.
Thank you. Bye-bye.
Bye.
Thanks for joining us, everybody. We'll see you next month.